Hi, everyone. This morning we have Takeru Honda from Tokyo Institute of Medical Science with us. Thank you so much for being with us, Takeru. Takeru is an engineer who has been describing models and measuring activity of behavioral work in people with cerebellar damage. And he's going to describe for us the ideas that he published about 4 years ago in PNAS. Describing models of cerebellar formation. As you know, many of us think about the cerebellum as having something to do with learning internal models. Whether it's a forward model or an inverse model is unclear, and I think his work is going to clarify that for us today. Takeru, thank you so much. Please go ahead.
Thank you very much. Good morning, everyone. Thank you very much, Professor Shadmehr, for your kind invitation, and thank you very much for giving me a precious opportunity. I have 3 sons, and now it is 11 o'clock p.m. in Japan. They are asleep, but someone may wake up and come here. Moreover, my network may be unstable. Please forgive me if these situations occur. Okay, let's start. I am Takeru Honda. Nice to meet you. I am Japanese. My background is computer science. My major is computational neuroscience. I study about the cerebellum and collaborated with Dr. Tadashi Yamazaki, Dr. Soichi Nagao, and Dr. Masao Ito. 53 years ago, the cerebellum as a neural machine written by Drs. Eggers, Ito, and Sentagotai was published. This book helped to clarify the cerebral circuit. In the cerebellum, input signal is conveyed to granule cells through mossy fibers. And granule cells convey signals to Purkinje cells through parallel fibers. Other input signal is conveyed to Purkinje cells through climbing fibers. Purkinje cells output signal from the cerebellum. These circuits are involved in some essential information processing. Sir John Eggers held the international symposium titled Information Processing in the Cerebellum. Dr. Masao Ito told me that this symposium was the best because researchers freely discussed. With Dr. Masao Ito, I summarized this story on this book. This was published in the 70th anniversary of David Marr's birth. In theoretical side, Dr. David Marr predicted the existence of plasticity between parallel fibers and Purkinje cells on the basis of the simple perceptron theory. Dr. Masao Ito researched VOR by using rabbits and provided Procur's hypothesis. Furthermore, Dr. Masao Ito found long-term depression LTD. Therefore, it is possible that the cerebellum functions as a learning machine. Here we have a question. What does the cerebellum learn? The cerebellum controls movement of muscles. We hypothesize that this motor control consists of the controls of 2 distinct quantities. They are gain and timing. Gain control means amplitude of movements. Timing control means the onset and offset of movements. We focus on timing control in this presentation. Timing control has been intensively studied using an experimental paradigm called Pavlovian eyeblink conditioning. In Pavlovian eyeblink conditioning, an animal is exposed to paired presentation of a situation, sustained tone, and an air puff that induces an eyeblink reflex. Tone air puff stimulus presentation with a fixed interstimulus interval between tone and air puff onsets is repeated. Look like this. After this conditioning, only a tone is presented. The animal has learned to close its eyes in response to the tone at a specific time, even if an air puff is not fed. When the cerebellum is removed, the animal cannot learn to close its eyes with an appropriate timing. Therefore, the cerebellum memorizes the interstimulus interval between tone and air puff stimuli onset. Next, I will explain these mechanisms with the structure of cerebellum. A tone stimulus is conveyed to granule cells through mossy fibers. Granule cells excite Golgi cells and Purkinje cells through parallel fibers. Golgi cells inhibit granule cells. When an air puff stimulus is presented, this stimulus is conveyed to Purkinje cells through climbing fibers. The Purkinje cell output an eyeblink reflex signal. Here, I'll introduce one of the experiments data. Dr. Hesro's group used to decerebrate ferrets. They directly stimulated mossy fibers as a tone stimulus, and they also directly stimulated climbing fibers as an air puff stimulus. During paired stimulation, the ISIs between CS and US onset were 200 milliseconds and 500 milliseconds in these examples. Extracellular signal unit recordings were made from Purkinje cells. The figures show the PSTH of Purkinje cells. Abscissa and ownage represent time and firing rate of Purkinje cells, respectively. They found that Purkinje cells stopped firing here when only mossy fibers were stimulated after conditioning. Then, if the cerebellum runs time, the passage of time needs to be represented somewhere in the cerebellum. Dr. Yamazaki and Dr. Tanaka hypothesized that the passage of time is represented in the cerebellar granular layer, and they built a computation model. To explain these mechanisms, we built a realistic computational model in the basis of experimental and anatomical data and attempt to reproduce results of animal behaviors. In this study, we used a neuron simulator genesis. All model cells use Hodgkin-Huxley equation. To be concrete, we built a two-dimensional layers and set connections from model Golgi cells to granule cells at random. We also built a multi-compartment model composed of soma and a dendrite and added a voltage gate NMDA channels to the dendrite. I show you our simulation results. This panel represents a spike patterns of 30 out of 1,024 granule cells. In our simulation, granule cells randomly fire when input signals are presented to granule cells at 1 second. You can see population of active granule cells exhibited random alternations between burst and silent, bursts and silent. Each granule cell exhibits random alternation between burst and silent modes. For example, granule cells E shows a random alternations between burst and silent states, looking like this. I focus on this pattern. This pattern is similar to near patterns, here, here. But this pattern is different from this pattern. Thus, different granule cells exhibited different temporal activity patterns so that the population of active granule cells changes gradually with time from the CS onset without a recurrence. When this is true, one population is assigned one time, looking like this. We hypothesize that granule cells represent a passage of time. This is a result in our simulation. Now I focus on this pattern. This pattern is similar to this pattern, but this pattern is different from this pattern. We calculated similarity index in order to determine whether the same active granule cell populations appear more than once. To confirm this non-recurrence property, we calculated the similarity index by using this equation. You can see that the index value was 1 at tau equals 0 millisecond. Because of the trivial identity, the similarity index monotonically decreased as the value of tau is changes. Thus, population of active granule cells can represent the passage of time. To examine the importance of simulated NMDA channels at our model's Golgi cells, first, we blocked the channel and carried out a simulation. These panels represent the results when the CS signals was presented. Population of active granule cells did not exhibit random alternation between burst and silent states, and granule cells elicited spikes stochastically. Similarity index value of spike patterns of granule cells in response to a CS maintained a high level compared with default case. This indicates that when NMDA channels are blocked, granule cells do not exhibit POT passage of time, and NMDA channels are necessary for the POT passage of time representation. NMDA channels are necessary for the passage of time representation. Finally, let me explain simulation of delay eyeblink conditioning. This panel represents a schematic of our delay eyeblink conditioning model. As shown, current was injected into granule cell. As air path, we conveyed the timing. We regarded the pause of Purkinje cell firing as an eyeblink. Tone signals are conveyed to granule cells. Granule cell represents the passage of time. Look like this. Purkinje cell receives signals from granule cell. Also, at this timing, air path signals are conveyed to Purkinje cell through priming fibers repeatedly. Because this is repeated, LTD is occurred at this timing. Thus, Purkinje cell cannot fire at this timing. We conducted simulation of delay eyeblink conditioning model. The abscissa and ordinary represent time and membrane potential of Purkinje cell, respectively. US signal was assumed to be fed to Purkinje cell 0.75 seconds after the CS onset at the time of conditioning. The Purkinje cell stopped firing around 0.75 seconds, indicating the eyeblink is occurred at this timing. This figure shows the firing rate of Purkinje cell. In this simulation, US was presented at 0.65. Our simulation showed reproduced experiment data of Dr. Hessel's group. There are activity patterns of granule cells. We can change the parameters of input stimuli and magnesium ions concentration of NMDA channels on Golgi cells. Granule cells show passage of time in red area. Granule cells also show synchronized activity in green area. When animals are silent, granule cells simultaneously activate. When tone signals are presented, granule cells represent passage of time. Look like this. Summary. We propose a computational model of the cerebral granular layer. Population of active granule cell represented the passage of time. We found the dynamics of model granule cells activity change with the extracellular concentration of magnesium ions and input current strength. Our cerebellum model reproduced eyeblink conditioning experiments.
Thank you. May I ask a question?
Yeah. Okay.
Maybe now is a good time to let people, if they have questions. I'll just ask the first one. Do complex spikes respond to the onset of the tone as well as the air puff? And if so, if the complex spikes respond to both, how does the cerebellum use the complex spike information to learn only with respect to the unconditioned stimulus?
I see. Of course, so CS conditioned stimulus is a tone or electrical shock or some stimulus. Of course, climbing fiber signal is conveyed to Purkinje about tone stimulus or air puff signals or electrical stimulus. It is a very difficult question. How does the cerebellum, the brain or human animals can detect between these stimulus, right? Actually, I don't know. I have no idea. But cerebellum is the same as a liquid state machine. A liquid state machine have ability of distinct information. So information A is very similar to information B, but the cerebellum can understand this is this signal is A or this signal is B because granule cell, the number of granule cell is very large, so granule cells can represent the information of detailed information. So maybe granule cells can represent stimulus A or stimulus B. But I don't know. Climbing fiber can understand this is signal A or signal B. I'm sorry, I have no idea.
That's fine, thank you.
So actually, Dr. Masao Ito said that this is 7 problems, 7 questions. So one is one of 7 questions is what information climbing fiber understand? Is it okay? Thank you very much. Okay. Any questions?
Hello. I think I
Hello.
didn't really get the burst pulse activity pattern of the granule cell. So the NMDA receptor is on the granule cell or Golgi cell? Like
Actually, granule cells have NMDA channels, but NMDA channels of granule cells type is different from Golgi and NMDA channels of Golgi cells. So NMDA channels of Golgi cells has magnesium ions, so they can block the ions, potassium ions or sodium ions.
Okay, so you blocked NMDA channel on Golgi cell?
Golgi cells.
So reduce the inhibition. No, block NMDA means reduce the Golgi cell activity. So
Actually,
it increase the granule cell activity.
Right. But Golgi cells have AMPA receptors, so they can receive the AMPA receptors input. Okay, so point is the time windows. So AMPA receptors, in AMPA receptors case, time window is very narrow, but NMDA channels of Golgi cells, time window is very broad.
Time window for what? Sorry, time window for receiving, responding photon.
No. One signal is action potential is here and down. So this this time window.
Action potential of from input
Yeah.
Golgi cell, from granule cell.
Yeah. Golgi cells.
Golgi cell. So I think my confusion is what. So burst pulse pattern is important for something, and NMDA receptor is critical for making that burst pulse pattern.
Right. But
okay. So
sorry.
What's important for this burst pulse pattern? Is that important for learning?
No. This is very important for making a pattern of active granule cells. Passage of time. Yeah.
So if the cell reads that pattern, what happens?
Wait.
Sorry,
I don't have a slide, so I seek slides here now. Oh my God, where? Nothing. Sorry. Wait a minute. Sorry. I mistake. Wait a minute. I'm sorry. I don't get the slide. Sorry.
No, never mind. Just I get back to you later sometime. So please go ahead.
Okay. Hi, thank you very much. I'm sorry. So I continue. In 1958, sorry. In 1958, 1958, Dr. Rosenblatt provided an idea of the simple perceptron. Dr. Mass provided an idea of the liquid state machine. The liquid state machine is the same as our model. AI technology is developing these days. Most recently, liquid state machines have been called reservoir computing, and attempts have been made to apply them to AI. Acknowledgements. And next, here we came back to this question. This is one idea. The cerebellum learns internal models, which play an important role in motor control. Dr. Masao Ito focused on cerebellar loops. He came up with an internal model hypothesis, especially forward model. Cerebellum learns internal models, which plays an important role in motor control. We held Fujiwara Seminar 2018 in Tokyo, Japan. Many cerebellar scientists joined this. Tokyo Declaration was presented. In this declaration, there is an urgent need to better understand the healthy cerebellum and the mechanisms that lead to cerebral disease, and close partnership between scientists, physicians, patient organizations, and funding agencies are very important. Thus, in study fields of the cerebellum, the collaboration is very important. Next, I would like to introduce a discovery in clinical side. In clinical practice, physicians subjectively estimate SARA score. As shown in this figure, we can see up and down of SARA score. However, SARA can comprehensively score motor cerebral ataxias. Dr. Tom Such group researched learning of gaze slowing direction. When healthy subjects wear the prism goggles, their view field shift leftward. So they could not hit the target after around 15 trials. A healthy subject hits the target. This is adaptation. However, a patient with inferior rib hypertrophy could not hit the target and could not adapt the prisms. Look like this. Therefore, in clinical side, it is suggested that the cerebellum functions as a learning machine. Next, I would like to introduce an example of collaboration on the basis of learning hypothesis. We developed a measurement applying the prism adaptation task. When subjects touch the ear sensor, the target appears on the touch screen. Subject's visual information deviates leftward in this case from the effect of prism. When subjects release their finger from the ear sensor, they cannot see the target and their hand because the target disappears and the electronic shutter on the goggles closes. When subjects touch the touch screen, this horizontal displacement is measured. Subjects repeated this task, look like this. After many tries, subjects can precisely touch the target. We developed the adaptability index AI. We focus on the last 10 trials of wearing prisms. The initial 5 trials of removal of prisms and the last 10 trials of removal of prisms. The probability of correct touch or incorrect touch is calculated. AI defined this. In this healthy subject's case, AI is 1. This is a healthy subject case. AI value is 1. But patient with spinocerebellar ataxia 6, AI value is 0.012. or a patient with SCS6. In this case, AI is 0.0. They can touch the target, but when goggles were removable, they can touch the target, so memory is disappeared. Or this patient with SCA spinocerebellar ataxia 31, AI value is 0.144. They can touch the target, but they couldn't touch the target, but they cannot adapt. Look like this. We evaluate the volume of cerebellum. In adults case, right lobule 6 and left crus 1 correlate with AI. AI is a new clinical index for the quantification of cerebral motor learning. Next, we developed a system which can objectively measure a SARA score. This is a part of SARA, finger trace, rated separately for each side. Our system, including Kinect V2, detects movement of trunk, arm, and so on. In our system, a white circle moves. Subjects have to move a black circle and insert the black circle into the white circle. We analyze the distance between target and a subject index finger. Red and blue show target and subject's index finger, respectively. The error of the patient is larger than that of healthy subject. In SARA, physicians have to measure the distance, 5 centimeter, 50 centimeter, and so on. However, it is difficult to measure it objectively. Our system can do it. Fortunately, it is possible that data obtained from our system can be compared. Children, adults, patients, dystonia patients, cerebral patient, dystonia patient, Purkinje disease patient, aging, and so on. Finally, I would like to briefly introduce finding in basic science side. In basic science, there is one question: what information does the internal model represent in the cerebellum? In the case of offline feedback, subjects confirm the target with their hand when they touch the screen. In the case of no feedback, they cannot see the in the case of no feedback case, they cannot see the target and their hand when they touch the screen. We thought that state of internal model is revealed by the no feedback case. In our task, the no feedback condition was used as a catch trial. These are the results of prism adaptation task. In cases where no prism were worn, healthy subjects precisely touch the target. After putting on the prism, they could not touch the target during no feedback. But these horizontal displacements quickly decrease during offline feedback. During no feedback case, they precisely touch the target. It is suggesting that they adapted to the prism effect. Comparison of these points show motor memories act as an internal model. In the case of cerebral patients, it is not significant difference between these points. It shows that internal model is constantly updated during offline feedback condition. There are two ideas about internal models. Which internal model is represented in the prism adaptation task? The forward model or the inverse model? The healthy subject repeated alternating between 10 trials of offline feedback and 5 trials of no feedback. The black and red dots show the offline feedback and the no feedback cases, respectively. They rapidly became able to touch the targets in the offline feedback case correctly. This is fast adaptation. On the other hand, it took many more trials to correctly touch the targets in the no feedback case. This is slow adaptation. We can see the internal model observed in the no feedback case changes gradually. Slow adaptation is hidden in fast adaptation. At the beginning of the prism adaptation task, healthy subjects do 10 trials in the no feedback case. I assume that this deviation is the baseline. This is Y, this is X. I got D equals Y plus X. Therefore, fast adaptation consists of X and slow adaptation. This suggests that X and Y represent different internal models, mechanisms of adaptation in the cerebellum. Next, participants were instructed to intentionally miss the target. We call this non-aimed offline feedback task. In this task, we do not find the adaptation observed in the offline feedback task. Since teaching signal is important, internal model observed in no feedback condition represents inverse model. Y, which is slow adaptation, represents inverse model. Next, participants touch the target precisely at the beginning of offline feedback task after 30 trials of no feedback offline feedback task. Participants could not touch the target at the beginning of offline feedback task after 30 trials of no feedback task. Therefore, since visual error signal is important for learning in non-aimed offline feedback condition, fast adaptation also includes forward model. X represents forward model. Here, I try to combine two ideas and provide tandem forward and inverse models. Y represents the output of the feed-forward circuit, including the inverse model. X represents the output of the internal feedback circuit, including the forward model. Therefore, I also go to D equals Y plus X. We calculate the gain of inverse and forward models. We estimate the activity of forward model and inverse models. So tandem hypothesis could be confirmed if plasticity between parallel fibers and Purkinje is measured during learning. We conducted simulation of impairment of the inverse model. During offline feedback case, horizontal displacements quickly decrease. However, during no feedback case, horizontal displacements do not decrease. We found the same phenomena of cerebellar patients. Next, we conducted simulation of impairment of the forward model. During offline feedback and no feedback cases, horizontal displacements do not decrease. We found the same phenomena of cerebellar patients. Look like this. Tandem hypothesis explains the behaviors of cerebellar patients. Cerebellar patients are impaired in the either forward or inverse models. Summary. Clinical side, adaptability index AI represents a capability of cerebral motor learning. Kinect system quantitatively evaluates SARA scores. Cerebellar patients exhibited impairments of either forward model or inverse model. Basic side, the cerebral internal model combines the forward and inverse models. Tandem models display the fast and slow adaptation in learning. Acknowledgement. Thank you for your attention. Thank you very much.
My pleasure, Takeru. Thank you very much. Let's see.
Thank you very much.
Let's see if there are some questions for you. I will start. So you dissociated forward and inverse models, if I understood it correctly, based on the rate changes that were taking place, and other ones. It's possible that there are fast and slow mechanisms of learning that contribute to adaptation. How do you dissociate the content of the internal models, what is being learned, from the rates with which those are being learned?
Okay. So, slow adaptation case. So actually, in no feedback case, participants cannot learn about deviate the prisms, right? So point is offline feedback. They can understand the error, and they try to touch the target, and they want to modify the movement, right? In this situation, they can learn about forward model and inverse model. So why is the slow adaptation is why does the slow adaptation occur? So because in offline feedback case, they can participants learn forward model and inverse models. In no feedback task, they can represent motor control by using the inverse model. So this is a result of inverse model in slow adaptation. Is it answer for it? Is it the answer of you?
Thank you.
Question?
Yeah. Are there other questions for Takeru?
Yeah. Thank you.
Well, Takeru, sounds. I'm not sure. Are there any other questions? Thank you so much, Takeru, for taking the time to present your work. I very much enjoyed the model that you presented at the beginning that described the passage of time with the granule cells.
Yeah. Thank you very much.
My pleasure. Have a good
Thank you.
Have a good Tuesday, everybody. Thank you. Goodbye.
Thank you very much. So I want, oh my God. Thank you very much.